Welcome. In today's lesson, we want to continue on the movement of substances. So last time when we talked about the movement of substances, we talked of one thing called diffusion, right? So, and when we looked at diffusion, we said that diffusion yedu is the movement of particles from a region of higher concentration to a region of low concentration. So now what we want to look at is just a continuation here diffusion yedu, but in this continuation of diffusion we are primarily looking at a specific molecule. That specific molecule iroro rinonzi water. So when we talk of osmosis, we are saying that osmo anoreva kuti water, and osmosis will be the movement of water, but it is a diffusion of water because osmosis is a type of diffusion. So let's look at our first note. All cells are surrounded by the cell membrane, which is partially permeable. And we talked about it. We said that partially permeable, it means it allows some other substances to enter through and other substances have mizo. Then water can move in and out of cells by osmosis. Osmosis, by definition, is the diffusion of water. As I had said earlier on, that osmosis is a type of what? Diffusion. So osmosis is the diffusion of water molecules from a dilute solution. A dilute solution yedu ndio inenge in a high concentration of water to a more concentrated solution, which is low concentration of water across a partially permeable membrane. Saka tine about three things or three parts that we must get there. So maybe I use another color. Tukuti osmosis yedu, it's a diffusion, right? So by diffusion we say you are moving from a region of high concentration to a region of what? Low concentration. Is that okay? Then this is the diffusion of water molecules, and so water molecules are from a region of high concentration to a region of low concentration, and it. So if we are to say osmosis is the diffusion of water molecules from a from a region of high concentration of water to a low concentration, to a region of low concentration of water across a partially permeable membrane. So that is your primary definition, and it. But kwatrukuti a region of high concentration of water and that type of a solution is called a dilute solution. So let's say for example you drink rako ukari diluta uri kuedera water. So water concentration inenge yakawanda mu a dilute what? Solution. Then to a more concentrated solution. So concentrated solution yedu inenge ine less of water, ndo yatiri kuti drink redu risati radilutwa harinachi mvura inenge irimo yakawanda. So which means drink rakadilutwa rine a low concentration of water but a high concentration of the solution, and it, but low concentration of water. Then drink redu rakadilutwa rinenge rine high concentration of what? Of water. So when you look at osmosis, you are primarily looking at the movement of the water, not of the solution. It's of the water. So water would move from a dilute solution which has a high concentration of water to a more concentrated solution which has low concentration of water across a partially permeable membrane. Then we go to another point. In doing this, water is moving down its concentration gradient. So it's moving down its concentration gradient. We are saying kuti more concentrated ndizvo more concentrated ndo patiri kuti inenge richi a dilute solution, then richibva raenda uko kune less concentrated, which is a what concentrated solution. Are we together? So it would move what down the concentration gradient, but down the concentration gradient of water. So water is moving down its concentration gradient, and then the cell membrane is partially permeable, which means it allows small molecules like water through, but not larger molecules like solid molecules. So the cell membrane only allows water, and it doesn't allow the other molecules to pass through because of their size. They will be larger. So let's try to look at the analogy here of osmosis and the partially permeable membrane. So here we have a partially permeable membrane yedu, which is this part. That's the partially permeable membrane. Saka membrane redu rine zvimaburi zvanditi. Then on our membrane we have we do have what molecules, right? So we have large solid molecules, which are these ones, a red eye. Are we together? So these large solid molecules, a red eye, kuti anonopinda achinonopfuura netumaburi ututu, anonopinda achinopfuura tumaburi ututu, they can't do that. Saka they can't do that primarily because of their what? Of their size. So the large molecules are unable to pass through the membrane. Then the other smaller molecules are blue ututu, right? Blue ututu. Tunoti tukanonopinda nemaburi iwawa tunoita sei? Tunopasa through maburi iwawa, right? Saka tukupasa through maburi iwawa. Why? Because they are smaller than the what? Than the maburi achona aripo. And so the smaller water molecules are able to pass through the membrane. So ndo yatiri kuti partial permeability kana kuti partially permeable membrane. So after having known osmosis, what it is, we have to look at theory behind it. Its theory behind. We want to discuss osmosis, maybe particularly we're explaining a particular factor, and then we want to consider osmosis being the one here true chance to explain that factor. We would have to look at the theory of osmosis. And so the theory will suggest that osmosis is the net movement of water molecules. So pekutanga papa, we just only talked about it, be defining it as the movement of what? Water molecules from a region of high concentration to a region of low concentration. But here now we are talking of it as a net movement of water molecules. A net movement of water molecules. Are we together? Saka tukureva kuti in overall ma molecules edu achazoenda which side ndizvo. Let's say for example we have a membrane, and tine membrane, both sides of the membrane kunze kwecell nemukati mecell mese munenge mune mvura, anditi? But mune mvura kudai, so titese tichazoona kuti pane kuchapedzera kwazoenda mvura, titese kunoenda mvura yakawanda, because kwese ichabuda ichapinda ichabuda ichapinda, but yatinozoti net ndeyekuti kwainonyazonyanyowandira kuenda ndekupi. Are we together? So the osmosis here is the net movement of water molecules from a region of higher water potential. So we are introduced to another term, higher water potential. Water potential being what? It's being the ability of the water to move to treat it. So let's say, for example, you have a dilute solution, right? Dilute solution in a concentration yakawanda ishi water. So which means it's easy for water to come out of the dilute solution, to come out of the what? Dilute solution, to move away from the dilute what? Solution, because its concentration yakata say yakawanda. And it so a highly concentrated area ye water, we say that that area inenge in a high water potential. So that's the dilute solution with high water potential. So apa runs to a region, saka the net movement from a region of high water potential to a region of lower water potential. Lower water potential ndo kwatukutaura kuti kune mvura shoma kwachona. That's the concentrated what? Solution. And through a partially permeable membrane. So as you can see, by theory we are now trying to make it very easier kuti munhu pamwe probably we are going to talk about the concentration of water, tobva solids nemachi. So the more yatinowanza terms ekuti concentration ndizvo sometimes pamwe munhu anobva aita confusion kuti concentration iyi yambova yechi andita. So rather would make use of the word what? Higher water potential, lower water potential. Higher water potential is what? Higher, that's where a lot of water is. Higher water potential to a region of lower water potential to a region where water is short-minded. Then on our second point, it can get a little confusing to talk about the concentration of water when we also talk about the solutions being concentrated, having a lot of solute in them. So instead, we can say that dilute solution has a high water potential. Then the right hand side of the diagram below, and the concentrated solution has low water potential on the left hand. So we want to look at the diagram, and the diagram read right below is next to this. And now as you can see on the diagram, toda kuona pane high water potential and low water potential, and it so we are saying high water potential, not necessarily to say where there is a greater volume, and it but we are saying where there is potential of the water molecules to move away from, and it so let's look at this diagram. So panapa, as you can see, we have this volume and this volume, and as you can see, volume here per 1 and volume here per 2 apapa ndirere. Volume here per 2 yedu ishoma, then volume here per 1 yakawanda. Ukangotashandisa zvekuti kune volume yakawanda ndokunoita, kune volume yakawanda ndokunoita, unokwanisa kutadza, anditi? Because apa unokwanisa kutofunga kuti water would have to move kubva ku 1 ichienda kuchi? Ichienda ku 2, anditi? Of watopa reason yako, because there is a high what? There is a greater volume of what? Of water at 1 and a lesser value or lesser volume of water at 2, and it. But when we talk about the water potential, we're not merely talking about the volume of what? The water. We are talking about how water in a potential ya yipi yekubuda treatise. So in other words, we are going to talk about it's what the concentration yes solution. Is it a high concentration or it's a dilute concentration? And it dilute concentration in a drink shoma, in a solution shoma. Then highly concentrated in a solution yakawanda. So as we can see with these two parts, right, we find out that this side imaging 1, 2, 3, 4, 5, 6, 7, 8. So we have these red guys, 8 of them is solid ndiro red. Saka kana kune 8 is solid ndiro red, then this part kune how many? Kune 2. So which means to say that this is what? This solution is what? More concentrated. And so kune concentrated what? Solution. Then kunoku ku this side kune 2 kune dilute solution. Saka since zvadai, so now we would want to know the net movement. And so by definition yedu, or by theory yedu, we say osmosis is the movement of water molecules from a region of a high water potential, which is a dilute solution, to a region of low water potential, which is a concentrated solution. So water yedu, H2O, is going to move from this end to this end. So we find that the net movement will be this movement. Are we together? So that's the net what? Movement of what? Water yedu. Are we together? Then we go to this part where we have to look at it in terms of the concentration gradient. So kana pane net movement, and we will be saying it will be from higher water potential to lower water potential, but down a what? A concentration gradient. And it saka kubva uku kwayabva kunoku kune side rine 2 ichienda kuside rine kuside kwakanzi 1 ukuku, it is moving from a region of high water potential to a region of low water potential. So always keep it in mind kuti pese paunoona water potential, just don't think of it as a greater volume. Just don't think of it as a greater volume. Think of it as a greater concentration. In other words, you have to think of it as a dilute solution. A dilute solution has higher water potential than a concentrated solution. It has lower water potential. So water would move from a region of high water potential, which is the dilute solution, to a region of low water potential, which is the concentrated solution. So we have an exam tip. So most times when you talk about osmosis, if you refer to water potential, you'd understand your concept what better, and your explanations become better. Are we together? Other than utangotaura kuti the concentration of water, the concentration of water, the concentration of the solution, the concentration of concentration anowanda, after awanda, at the end of the day hazvichanzwisisiki pamwe iwo you'll be the first one anototadza kutozonzwisisa your concept. And then we go further ne-osmosis yedu. So since we have known the theory of it, let's try to look at the practical part of it and how it is used in what? In experiments. And so our first point is that the most common osmosis practical involves cutting cylinders of potato, placing them into distilled water and sucrose solutions of increasing concentration. So we have distilled water. Distilled water, we just distilled water uku kune high what? Water what? Potential. Then uku kune low water what? Potential. Ndo maonero anenge uchizviita handiti. Saka in other words they are saying tine potato cylinders edu, tichamaisa rimwe ticharisa mune high water potential, then rimwe ticharisa mune low water potential. And then the potato cylinders are weighed before placing them into these solutions. So we're saying kuti tamaweya, we want to see a change, right? So tikatora mass ye product tisina kutora mass ye before paitika anything, we would be able to what? To compare. And so that mass yatinotora pekutanga iyo, that initial mass after cutting the potatoes edu, it's so that we can compare kuti pawedzera here iyi inonzi mass here, oro hapana kuwedzera mass, oro padecrease mass, such that we can determine what has happened as an effect. And then they are left in the solutions for 20 to 30 minutes and then removed, dried to remove excess liquid. so yatiri kuti dried to remove excess liquid here kungopukutwa hakusi kuti aomeswa and then they are reweighed. Are we together? Then after aitwa reweighed ndizvozve would have to expect to see my differences in my masses, either imwe yakagaina or imwe yakaita se yakalose mass. So the potato cylinder in the distilled water, distilled water, and remember distilled water, we said that the distilled water ndo in a high water potential. So the potato cylinder in the high water potential will have an increase in its mass. Saka yaita an increase in its mass the most, it's because water would move from a high water potential distilled water into the potato rinenge rane a lower water potential. Saka water inoitwa draw in into the potato cylinder. Then the potato cylinder in the strongest sucrose solution. So our sucrose solution, remember, that's where we have low water potential, will have a decrease in its mass the most. So we are saying that potato cylinder redu raiswa mune sucrose, and mune sucrose umomo there is low water potential, so water is needed in sucrose, saka ichatosvetwa away from the what, from the potato. Saka potato redu parinotorerwa mvura richisvetwa ndo patinobva taita a decreased what, mass. Are we together? So if a potato cylinder that has not increased or decreased mass, if there is a potato cylinder, it's not going to decrease a mass, it's not anything. It means that there was a balance, right? So it means that my concentrations, actually my water potentials anenge akaita se akafanana, kuti mumvura mune water potential yakafanana nemupotato, saka hapaitike any network movement, and it. So on our note here, if there is a potato cylinder that is not increased or decreased in mass, it means there was no overall movement of water into or out of the potato cells. Okay, let's see. Okay, let's see if there is a difference. Okay, so the most common osmosis practical, okay, is cutting potato cylinder. I think that sound is a repetition. So this means that more water molecules will move into the potato cells by osmosis, and osmosis will cause what? Water to move into the potato, pushing the membrane against the wall and also increasing the Tagger pressure in the cells and making them what? Turgid. So it's also tachitaura kuti if osmosis takes place and then you find out that paitika a net movement yekuti potato ra increase in what, in its mass, and it chinenge chaitika ndechekuti tikuti mvura yedu yaita sei, yapinda mukati mandi yapinda mukati umu ichapfurira ichinonopinda umu titese, then yapinda umu ndizvozvo here ichawanda because iri kukweviwa ndizvozvo here, yawanda ichikweviwa ndizvozvo here, then that would exert a what, a pressure here. Are we together? That would exert a what, a pressure. Then that pressure inoexertwa iyoyo is called what, Tagger pressure. So Tagger pressure is the pressure that is exerted by the cell membrane against the cell wall. Cell membrane cell wall. That would be what, Tagger pressure, and it. Saka, in other words, we would call that cell a what, a turgid cell. And the potato cylinder in the strongest concentrated solution will have its mass what, decrease, and the most as there is a greater concentration gradient is the tube between the potato cells. So and sucrose solution. This means that more water molecules will move out of the potato by osmosis, making them flaccid and decreasing the mass of the potato cylinder. So isusu tikutaura kuti chii? Tikutaura kuti on the other hand pane potato futi raiswa mucylinder meya pane potato cylinder raiswa mune concentrated solution tritase, and that concentrated solution tachiti ine a lower water potential. So as a result, the net movement ndeyekuti water will be drawn out of the potato cell and it into the solution, and it so draw out, so you find out that water moves out, right? Water moves out, and it saka water iri mukati yamove out mukati muchapedzisira makuita sei raakudhonzeka. Are we together? So you find that the cell membrane riri kuita richase richidhonzeka, and this cell membrane ndirozve it goes away from the cell wall. Saka tikuti this one is what? The cell wall. This one is what? Cell membrane, and it so you would find out that the water dzinonzi because mvura yashoma ndizvozve yakashoma muno, the cell wall would move towards the side, and it ramove towards the side richidhonzwa nekuda kwekuti mvura ikuita shoma. Cell membrane yedu yakubva yamove away from the cell wall, creating gaps utu twatinozoona utu tunoita kuti tikwanise kuzoona kuti that cell is what? Is plasmolysed. Is that okay? So the cell redu rinozonzi ririchi, it would feel floppy, and it is described as a what? As flaccid, and it so if looked at underneath the microscope, the cell from the potato cylinder might be plasmolysed, meaning the cell membrane has pulled away from the cell wall. So like a plasmolysed, it would mean that the cell membrane has been what? Pulled away from the cell wall, and it pulling away from the cell wall yatanzi iri kunzi iyochi plasmolysed. Are we together? Then if there is a potato cylinder that has not increased or decreased its mass, it means there was no overall net movement of water into or out of the potato cell. So this is because the solution that cylinder was in was the same concentration as the solution found in the cytoplasm of the potato cells. So there was no concentration gradient, and it so remember when we talked about concentration gradient, we said it is created when there is a difference within concentrations, and say kuti panapa pana 80 concentration yedu 800, then pana 20, we have a what? Gradient, and it but now if yekutanga ikaita 8 ndizvozve, then yechi2 concentration yoramba iri pachi 8 ndizvozve, so panapa would have a straight what? Line. That straight line inenge rine gradient of 0. Saka kana rine gradient of 0 takutaura kuti there is no what? No concentration what? Gradient. So that's what it merely means. And so after having known all this, we now want to understand the importance of osmosis in tissues, because osmosis is helping movement in what? Of molecules within the cells themselves. It is important primarily to what? To tissues. So previously, when we talked about cell differentiation, we also talked about the issue that when cells are of the same kind, they are of the same kind, they are grouped, a group of cells of the same kind becomes a tissue. So we're just merely saying that we want to look at the importance on overall ama cells akawanda are of the same kind. And so when water moves into a plant cell, the vacuole gets bigger, pushing the cell membrane against the cell wall. Are we together? Izvi zvavari kutiudza izvi ndo zvinhu zvokuitika izvo kuti inonzi mvura painotopinda muplant cell paipai chainotoitika ndechekuti the vacuole get bigger. Why is the vacuole getting bigger? Remember, the vacuole ndo yatakataura kuti ine mineral salts as well as what? Sugars. So in mineral salts and sugars, we are just talking of a sugar solution, a salt solution, and so that salt solution is concentrated. That concentrated part is the one that it kukweva ndizvo. So painokweva, the vacuole now becomes bigger. The vacuole yaita bigger, it then pushes the cell membrane against what? The cell wall. Then water entering the cells by osmosis make the cell rigid and firm. Are we together? Ndora tiri kuti ifootball iri mukati mune tube ndizvo. Tube ndo vacuole. Tube painozara ichizara inononoita press against what? The cell wall, which is chikoko chekunze. And ya press against that inobva yaita kuti paome chobva chaita rigid and firm. Then this is important for plants as it affects all cells in a plant being firm. Treatise is to provide support and strength for the plant. So cells akava turgid, chii chichaitika? Plant redu richaprovide what? Number 1, support, and number 2, richaprovide what? Structural strength. Are we together? So making the plant to stand upright with its leaves held out to catch the sunlight. Saka ukazoona plant redu rakanyatsomira zvakanaka rakabudisa leaves richishaina, inyaya yekuti panenge paitika chii? Turgidity. And then ukaona paipe parinodonha plant redu robva rarara, it's because excessive loss of water yaitwa titese. Saka the plant cells are no longer turgid. And then the pressure created by the cell wall stops too much water from entering and prevents the cell from bursting. So this cell wall, it is made up of what? Cellulose. And now this cellulose it's a structural sugar treated. It is structural sugar that structural sugar yakasimba treated. Yakasimba zvisingaite zvekuti nyangwe ikarapiwa pressure yakawanda ndizvo inoramba yakangoita stand. Saka nekuda kwekuti cellulose yakagadzira cell wall ndizvo yakonzeresa kuti cell wall rirambe rakaita raka-withstand pressure. That on its own, it prevents the cell from bursting. Because dai the cell wall risingakwanisa withstand that pressure, then the cell wall would then burst. Kana raputika zvinhu zvese zvobuda pasi. So if the plants do not receive enough water, the cells cannot remain rigid and firm, or even what we term to be turgid. And the plant then wilts. Kwava kuoma kwandi pabuda paita excessive loss of water, plant cell redu robva raoma, it wilts. Ndopaunoona kuti chi plant rinenge raneta radonha rikaramba richingotsva risina kunyatsoabsorba water zvakanaka, ndopatinozoona kuti zvinhu zvaya zvakatsva. Are we together? Then let's look at osmosis in tissues, primarily in plant tissues. So the plant cells are turgid. The plant cells that are turgid are full of water and contains a high turgor pressure. So when something is turgid, especially this plant cell, if it is turgid, it has a pressure that it has on its own. That pressure is called turgor pressure. So the pressure, turgor pressure is the pressure of the cytoplasm pushing against the cell wall. Then this pressure prevents any more water entering the cell by osmosis, even if it is in a solution that is a higher water potential than inside it. The cytoplasm, that inside the cytoplasm of the cells. Saka zvinei nekuti uchaisa kunhu kune higher water potential, as long as razara raita sei? Razara. Are we together? It's only kana risina kuzara parinokwanisa kuabsorba imwe more water by osmosis, nekuda kwekuti raiswa solution in a higher water potential. Then this prevents the plant cells from taking too much water and bursting. Then plant roots are surrounded by soil water. Plant roots are surrounded by soil water, and the cytoplasm of the root cells has a lower water potential than the soil water. So if it has a low water potential than the soil water, then the water in the soil would then have to go to a region of lower water potential that is inside the root cells. And so this means the water will move across the membrane of the root cells into the root by osmosis. The water moves across the root from cell to cell by osmosis again, and until it reaches the xylem. Once they enter the xylem, they are transported away from the root by transpiration stream. Are we together? Helping to maintain a concentration gradient between the root cells and xylem vessels. Are we together? So now, I'll show you chichaitika tanzi susu yabva kwese kwayabva from cell to cell yanga ichitransportwa neosmosis, handiti? Are we together? Yatransportwa neosmosis kubva kwese kwayabva koko, ndizvozve. Then hanzi ichingosvika kuxylem, once it reaches this xylem, are we together? Then yakunzi takupiwa another process yairi kutransport, hanzi it's transported away by what? By this process here, and the transpiration what? Stream. Are we together? So transpiration stream yedu chii chiri kuitika? When transpiration occurs, mvura inobva kumusoro kweplant kune mashizha kwaya, mvura inobuda. Then mvura yabuda kune mashizha kwaya ndizvozve kuchabva kwacreatwa kuti kune kwaane less concentration of what? Of water. And it then water yedu is then drawn, inopuliwa from kuzasi kweplant ichienda kumusoro kweplant inside the what? The xylem vessels. But the force yakudetermine the pull titese is no longer just a passive process iringoitika yega, but yakudonzwa, not necessarily say that it is a negative process. No, it's not a negative process. I'm trying to say kuti there is something that is facilitating the diffusion. I think that word facilitating the diffusion is better. And yakufacilitwa netranspiration pool. Then that transpiration pool yakukonzeresa kuti mvura itese idhonzwe kubva kuground ichita se against gravity, handitika ichikwidza ichinonoenda kumusoro kweplant kwainenge ichidiwa, handiti? Then we also have to look at the movement of substances in what? In animal tissues, handitika, and we look at osmosis in animal tissues. Animal cells also lose and gain water as a result of osmosis. Animal cells do not have supporting cell wall. They don't have a supporting cell wall. And remember, a cell wall, it consists of cellulose and chakaita chikoko chakaoma, ndizvo? Tochifunga se animal cell redu seballoon, bhalloon harikwanisa kucontrola kuti mweya wapinda wakwana, handiti? So which means that ukaramba uchifuridza bhalloon it will get to a breaking point, handiti? So the results, so let's continue. The results on the cells are more severe. In animal cells, if an animal cell is placed into a strong sugar solution with lower water potential than the cell, it loses water by osmosis and becomes cremated. Svinwa yobva unyana tritese becomes shriveled up, and it shriveled up ndo masawa aya. Masawa anoona unyana wakaita masawa anenge asvetwa mvura dzose dzabuda. And then if an animal cell is placed into distilled water with a higher water potential than the cell, then it will gain water by osmosis. As it has no cell wall to create tugger pressure, the uptake yewater iyoyo inoramba ichingocontinue until the cell membrane is stretched too far and it bursts. Animal cells, kana concentration, let's say kana water potential mairi kuiswa, water potential iri high, mvura ichatanga kutopinda mu animal cell redu, yapinda uncontrolled, hapana cell inokwanisa kucontrola, uncontrolled. Yaita uncontrolled inoramba ichizara until that cell gives up, robva rabesta. Are we together? Then let's look at this one. So pane important terms unofanira kuti ugare uchingoziva, such that there are terms that can be used to mainly describe those solutions atataura kuti ikaiswa mune high water potential, ikaiswa mune low water potential, and also ikaiswa mune potassium water potential. And so we have the first one, which is called a hypertonic solution, and it. So a hypertonic solution, particularly, that's where we have a solution in a lower water potential than the cells, than the water inenge iri mu cell munonzi mu cell vacuum, munonzi mu vacuum medu or mu cytoplasm medu. And so at the end of the day, cell redu ndo rinenge rine a higher water potential. Right, a higher water potential, water yedu ichabuda outside the cell. Yobuda, then those cells become cremated. And so as we can see, my red blood cells, for example, the red blood cells have a higher water potential than the solution. So as a result, the net movement of water is out. Then the cells become shriveled cells. Then we have another one, which is a hypotonic. We had a hypertonic and we now have hypotonic. So hypotonic solution is the one that is opposite hypertonic, and we are now saying that the water potential inside the outside the cells, that solution iyo inenge ine high water potential. In a high water potential ichakonzeresa kuti mvura ibve ukoko ichinonopinda mucell, handitika, because the cell rinenge rine a lower water potential. So for example here, the red blood cells have lower water potential than the solution. So the net movement is that water gets in. Then now once water gets in, atina a cell to control it is. Then to control by what? By pressuring a tugger pressure, ndizvo. Saka isina that cell chopedzera chaitika ndeti the cell swells up and it becomes what? Bursted. Titese. It may lyse. Are we together? Lyse, L-Y-S-E, means to burst. Then the last one is an isotonic solution. Isotonic. So isotonic, we're just saying that these guys are the same. water potential iri panapa, water potential iri kunoku, water potential iri kunoku. They are equal. So since they have an equal water potential, they will remain the same. And for example, the water potential is equal between the red blood cells and the solution. So there is no net movement of water and the cells are normal. So cells edu anofana kuita an isotonic what? Solution. They must have an isotonic solution. Are we together? Saka that sums up, I think it sums up what our discussion on what? On osmosis. And the next discussion we'll probably be looking at active transport. So that's it to osmosis.